guys, welcome to Shaan's Logic. This is me, Shaan, and I hope all of you are doing fine. Today we'll be doing a Science of Science episode. As I had mentioned in my previous episode, one of the topics that interests me are genetics and microorganisms, since it involves making something and modifying something at the cellular level. Today we'll be looking at what is genetic engineering, who are genetic engineers, how does genetic engineering work, where has genetic engineering been applied, what are the benefits of genetic engineering, what are some concerns around genetic engineering, and what are the common misconceptions around genetic engineering. Now, what is genetic engineering? Genetic engineering is changing the genetic material of an organism. This happens by removing, changing, or inserting a gene to make a recombinant DNA or rDNA. For those who don't know what gene is, a gene is a genetic material that codes for a particular protein in the body. We humans have been trying to manipulate the natural desired outcomes for various different reasons, whether it be healthy cattle or a fresh juicy apple. We have been performing selective hybridization and selective breeding for centuries now. Selective hybridization and selective breeding is nothing but letting healthy crop or cattle with desired characteristics to breed together. The outcome of this was sometimes we desired and sometimes not, as anything created by breeding has random characteristics. However, with genetic science, we can be sure of the characteristics we get with the crop or the cattle. Who are genetic engineers? Genetic engineers are highly trained professionals who use molecular tools and technologies to remove or add fragments of DNA, making a recombinant DNA. The overall goal is to remove or add genetic makeup for the better or to exchange the genetic material between species. How exactly does genetic engineering work? Genetic engineering is a very vast and complex topic. Let's look at a video that will help us understand how does genetic engineering take place.
Tomato. Doesn't this term sound a little weird? You might be wondering, is it a tomato or a potato plant? It's actually both. Yes, you heard me right. In this single plant, the tomatoes grow above the ground while the potatoes grow below the ground level. And how is this made possible? It can be done by a process called grafting. In this, the stems of both the tomato and the potato plants are chosen and then joined together by tying them. This is an example of plant engineering. On the similar lines, when we cut and join genetic material, the process is known as genetic engineering. It's one of the principles of biotechnology. Now let's explore more about these principles in detail. Most of the principles of biotechnology are based on two engineering branches, that is, genetic engineering and chemical engineering. Genetic engineering is the branch which deals with the modification of the genetic material of an organism. And which genetic materials are we referring to? We know that it's the DNA, which is the genetic material for most of the organisms. And what about the remaining ones? Well, in them, it's the RNA which acts as the genetic material. Just like in the case of some viruses. So genetic engineering deals with the manipulation of genetic material. Moving ahead with the next principle, which is chemical engineering. The name itself suggests that the principle revolves around the use of chemicals for the growth and the maintenance of cell cultures. For this, the desired eukaryotic or microbial cell cultures are stored in a contamination-free zone. Further, these cell cultures are used for the production of vaccines, antibodies, enzymes, and various other useful products. We will learn about chemical engineering in the upcoming videos. For now, let's just discuss in detail about genetic engineering. did you know that the first ever genetic modification was done in a bacterium? That's right. And later on, these modifications were applied to animal as well as plant cells. The genetic modification can be done either by adding something new to the existing genetic material, or it can also be done by deleting something from the existing genetic material. The DNA obtained after modification is known as recombinant DNA. Technically, genetic engineering can be done in many ways. Recombinant DNA is one way to pursue it. When genetic engineering is applied to modify organisms, they are known as genetically modified organisms, abbreviated as GMOs. And what organisms can be modified with this technique? Almost all living organisms can be modified with the help of genetic engineering. What kind of applications does this serve? Modifications in organisms can give rise to beneficial products. Consider plants, for example. With the help of genetic engineering, we can produce robust crops, pest-resistant varieties, and also high-yielding varieties of many crops. This was, in short, about one way in which genetic engineering can be carried out. The other way to perform genetic engineering is by making copies of the genetic material. This is also known as gene cloning. In simple words, gene cloning can be defined as the process of making an exact duplicate of the genetic material. But why do we need a duplicate of the genetic material? Let's consider an example to understand this concept. Imagine there's a new product launched by a company in the market. This has some really cool and amazing features which will be of great use to all. Now, is it fine if the company manufactures just one piece? Of course not. There will be so many people who will want to buy it. For this, there will be multiple copies of the same product manufactured on a large scale to suffice the demands. This just means that making exact copies of the original product is useful in several ways, isn't it? This same logic applies for genes as well. A few genes which code for certain extreme important proteins, or even the newly modified genes, can be copied several times. This process of making exact copies of genes is called gene cloning. And just as in this case, here as well, clones or copies of genes serve many important applications. This entire process of making copies of the genetic material falls under the umbrella of genetic engineering.
Where has genetic engineering been applied? Genetic engineering has been applied in various different fields like medicine, food production, crops, modified animals, etc. Let's look at a video on genetically modified meat.
Feeling hungry? Would you eat a steak grown in a laboratory? Would you take a bite of a cheeseburger made by a scientist? Technology startups and scientists are placing big bets on lab-grown meats. Currently, livestock is responsible for around 15% of the global greenhouse gas emissions. We could see this number rise in the coming decades. According to the World Economic Forum, the world population is expected to be close to 10 billion by 2050. That is a lot of mouths to feed. This population boom and rising incomes will cause demand for meat products to rise by as much as 88%. To feed the world's growing demand for meat, we already use around 27% of the world's arable land to raise livestock. We need to find a more sustainable way of satiating this rising demand. One path to the future of sustainable eating could start with lab-grown meats. Not to be confused with plant-based meat alternatives, lab-grown meats are cultured in the lab using cells which were originally derived from live animals. In theory, this cultured meat would use fewer of the planet's resources, and no animals need to be slaughtered in the process. Cultured meat might become a regular part of our diets in the coming years, making its way onto the dinner table and appearing in some of our favorite restaurants. Nonetheless, the prospect of this Meat 2.0 raises a lot of complex social, ethical, and technical questions. And, simply put, some people find the idea of eating meat from a lab weird. So what is lab-grown meat, and how is it made? First, a technician takes cells from a live animal. These cells are grown up in a lab to permanently establish a culture called a cell line. Once a good cell line has been established, a sample is introduced into a bioreactor. This is essentially a culture medium which contains all the materials the cells need to grow. The cells in the bioreactor grow and multiply exponentially and are then harvested. Once harvested, the meat cells can be formed into any number of unstructured items, from patties to sausages. In the future, it may be possible to use technologies such as 3D printing and edible scaffolds to create particular cuts of meat, like chicken legs and T-bone steak. However, this is likely to be much more difficult than producing ground meat. The technology's first public proof of concept appeared in 2013 in the Netherlands. Partially funded by Google co-founder Sergey Brin, Dr. Mark Post and his researchers at the University of Maastricht spent a little over $300,000 creating the world's first cultured meat hamburger. It was described as like an animal protein cake. The burger was even taste tested live on television and got positive reviews in general. However, the lab-grown burger lacked one central element: fat. In the meat that we eat every day, there are different types of muscles, connective tissues, lipids, bones, and fat that give flavor and texture to the food. To this day, one of the biggest challenges with lab-grown meat is giving them the flavors and textures that you might expect from biting into a steak or burger. However, this has not stopped entrepreneurs and scientists from trying. Today, startup companies are working to bring lab-grown beef, chicken, and even fish to dinner tables around the world. The cultured meat also needs to taste good or be close enough to more traditional dishes that the public won't mind that it comes from a lab. Until cell-based meat makes the leap from the lab to your dinner table, there are some delicious plant-based meat options you can consider that are gaining traction. Companies like beyondmeat and impossiblefoods offer customers meat-free options made from water, soy protein, coconut, sunflower oil, and natural flavorings. Some argue that the current appeal and adoption of plant-based meats by major restaurants could be a precursor to the coming age of cultured meat dishes.
What are the benefits of genetic engineering? First, let's look at the benefits of genetic engineering in the food industry. With the help of genetic engineering, food production can be enhanced, since genetic engineering can boost the yield of the food produced, as in it will have more quantity, and as well as it can help to increase the quality of the food, helping it to be more resistible. Benefits of genetic engineering in medicine. Genetic engineering in medicine can be used to create insulin, glucagon, human development hormone, birth-related hormones, etc. It can also be used to create different medicines, and it is also used to create vaccines for certain viruses. Genetic engineering can be used to improve crops. It helps the crops become more insect resistant and helps it survive even during the extreme conditions. It can also be used to modify animals and to create new species of animals, which increases the biodiversity. What concerns are related to genetic engineering? When you are genetically modifying a plant, you will have to give it more nutrients, more soil, more water, etc. However, this results in the downgrading of other species of plants surrounding it, since now it gets less nutrients, less water, less sunlight, less soil, etc. This leads to the other species of plants getting extinct, which harms the biodiversity of the world. If genetic engineering is not done rightly, it can poison the person who's taking the genetically modified product. What are the misconceptions around genetic engineering? Most of the misconceptions around genetic engineering are about the safety of genetically modified products of food, animals, etc. So, I want to say that if genetic engineering is done rightly, the products will do no harm. However, if there is some error with the genetic modification itself, of course it can do a lot of harm to the biodiversity, and if it is a food product, to the person who's consuming it. These were some things that I knew about genetics, and I'm still learning. I hope this video has been informative. Well, we have reached the end for this video today. I hope you guys like the video and will hit the like button. If you want me to improve on anything, please comment, share this video, subscribe to the channel, and I hope you have a nice and safe day. Sayonara.